Hey folks, in this video we're going to be talking about gene regulation, which is basically how you turn off and on the genes that determine which proteins you're making. Now, keep in mind we're going to be specifically talking about eukaryotes and I guess more specifically humans for today. And big picture with humans, we're talking about a huge genome. We've got per cell 46 chromosomes and more than 100 million base pairs per chromosome. So we're talking total, like 6 billion with a B, base pairs of DNA. And so I've drawn for you one partial chromosome here. And keep in mind when I'm drawing a single line like this, I actually do mean this is a double-stranded molecule of DNA. And this is just a tiny, tiny portion of 100 million base pair chromosome. All right, that's our starting point. Now, when it comes to regulating the genes, keeping them on or off or turning them on or off, the first and most important step is which genes do you even have accessible to you per cell. And this is determined in many ways by proteins called histones and how they're modified. So in general, we're talking about, are you wrapping up the DNA, making it really tightly wound so it's not accessible to various proteins, or if it's open and accessible and readable. Now, as you can read on the slide, what we can do is modify the histones, add little groups, little polar groups, non-polar groups, charged groups that change their protein properties that make them either bind to each other better or worse, bind to DNA better or worse, cluster tighter, etc., to just generally change the level of DNA condensation or tight wrapping, I guess. So if you add some of these groups, that tends to unwrap or unwind DNA, making it more accessible as seen here. And if you remove some of these groups, typically you get more tightly wound DNA. You don't have to memorize that, though. There's nothing specific here that you need to know. Most importantly, though, is understanding that which genes are accessible to you as a cell determine what kind of cell type you are. So if we have, say, 20-some thousand protein coding genes per cell in our body, maybe a skeletal muscle cell might use a specific 8,000 of those. A neuron might use a different 7,000. So which genes get wrapped up by histone modification, DNA condensation, is in many ways the prime determinant of what kind of a cell you are. All right, next up. The next thing to keep in mind is there are really important sequences of DNA that aren't even parts of the genes. Now, while our genes might be on the order of less than 2% of all of the DNA in our body, in our cells, these regulatory sequences, these other parts of our chromosomes that help regulate whether the genes are on and off, might be 10 times as much. So I'm not doing this to scale in any possible way. These are going to be grossly simplified. So in blue, green, and pink, we've got different sequences that are going to help determine whether the gene at the bottom of the screen are on or off. And the most important of these is this region called the promoter. And this is the particular location where, if you have the right transcription factors around, an RNA polymerase will be recruited. So remember, what we're talking about is a gene that's DNA, and if we can read that gene and make an RNA copy of it, then maybe we can read that RNA copy and make a protein, or hundreds of copies of a protein, as the case may be. So this is the promoter. And again, if we get an RNA polymerase there, we might make some RNAs and make some proteins eventually. So the proteins that bind at this promoter are transcription factors. And we've been talking about transcription factors for multiple units in this class. And these general or basal transcription factors bind to specific sites within this promoter or bind to each other. And that will, again, recruit the RNA polymerase. Now, here's where it gets a little bit tricky. Just because there is an RNA polymerase on the promoter right before a gene ready to go does not necessarily mean, despite that exclamation point there, does not necessarily mean that the gene's getting read. What you have to do is think about these other sequences in green and pink and the proteins that are bound there. This is the key. So, in addition to the transcription factors that bind at the promoter, there are also a wide variety of additional transcription factors that can bind at these other locations. I'm drawing a pretty fanciful, silly-looking set of transcription factors that are going to activate this RNA polymerase. Again, RNA polymerase by itself, just being there, isn't active. You could say that its on switch is there, but play hasn't been hit, I guess is the way I tend to think about it. And having these green activator transcription factors, as we call them, if they happen to be present, that is kind of like pressing the play button. They will perhaps covalently modify the RNA polymerase, stick it into drive, and get it moving. However, that may or may not only happen depending on if there are other proteins present or absent as well. These might be called repressor transcription factors, or more simply just repressors. And that's supposed to be a really bad drawing of a hand putting up a stop sign. And the idea here is it's the interplay between all of these other transcription factors that really determines whether or not an RNA polymerase is going to transcribe the gene, read the gene, or not. A couple additional clarifications. One is that all these DNA binding sites for these transcription factors aren't actually necessarily right next to the genes. They might be thousands, even millions of base pairs away. And if you want to know more about that, feel free to ask me after class, but not worth our time in this video. The other much more important level of detail to think about is going back to previous units. Remember that transcription factors are regulated, turned on and off, activated, inactivated by signal transduction pathways. So just remember what we're talking about here is which genes are available might determine what cell type you are, but your behavior on a moment-to-moment basis, for example, whether a muscle cell is contracting or not, whether a hormone-producing cell is releasing hormones or not, whether a digestive cell is releasing digestive enzymes or not, that's all determined by, perhaps hormones or signaling molecules bound to RTK or GPCR, receptor proteins activating kinases or producing second messengers and so forth, all the way down to the transcription factors again. So don't forget the big picture here that's harkening back to previous units about how signaling and understanding your conditions and what's happening outside the cell in your environment help determine what a cell is doing at any given time. Okay, let me clear that for now. Back to the one gene at hand. So let's look at this one gene and imagine what would happen if there were no repressor proteins or sufficient repressor proteins present, and there were activator transcription factors, and there were general transcription factors, and an RNA polymerase was recruited and ready to go. Well, it is going to do transcription, and that is to read this gene and produce an RNA copy of it. And so keep in mind this is a double strand of DNA. There are two strands. One strand is called the template. That's the one that's being read. But for today, I guess what I want you to focus in on is actually the coding strand, because if you read the template strand and make an RNA that binds to the template strand, it's going to look almost identical to the coding strand. You'll have to look at this on your own. I can walk you through it another time. But in a pre-mRNA, a transcript, is going to look almost identical to the coding strand, except that it'll be RNA. So that means, U's instead of T's, ribose instead of deoxyribose, etc. Okay, so here is our primary transcript, or just a raw RNA being made here. So as the RNA polymerase makes it to the end of the gene, you've got a pretty long RNA here. Keep in mind, polymerases always add to the 3' end, so that's the end that's being finished up right now, and the 5' end was the first thing that was made. So this right here is the pre-mRNA. It's not a fully mature mRNA, and we've got to do some processing. And there are 3 steps to fully process this in a eukaryote to make it into a mature mRNA. So the first of the 3 steps is to add a cap. So on the 5' end, we effectively take a GTP and stick it on backwards at the 5' end. You don't have to memorize that. It's nothing especially important. What it is just a very strange, unusual shape adding to the 5' end of this RNA. This will serve as kind of an exit visa to allow this RNA to leave the nucleus. Lots of RNAs don't leave the nucleus. This one will be allowed to if it has this 5' cap. The second thing that's done to this pre-mRNA is to add what's called a poly-A tail to the 3' end. So we're going to snip off the very end, and then a weird polymerase is going to add a bunch of A's. So just to the end of the RNA. And what this ends up helping is it helps keep the mRNA from being degraded. So when this RNA makes it out into the cytoplasm, proteins will bind to the cap and the tail, circularize the whole thing up, protect the ends, prevent our exonucleases, our antiviral machinery from chopping this thing up. Don't worry about the details. You can ignore everything I just said and just think this is protective. Our third processing step is to splice together some exons. Now, the basic concept here is that despite the fact that we just made a copy effectively of the coding strand of a gene, most of the gene is actually not that informative. Most of the gene is not actually instructions for making a protein. There are only short stretches that encode a protein, and these are called exons. There are intervening regions called introns that have to be eliminated. And there's more to it than this. There is a concept called alternative splicing that we can go over another time or you can look up. But for the purposes of this video, just accept that there are intervening regions called introns that have to be excised. The wording's very odd here. So introns are excised, they're cut out, while exons are spliced together and kept. So there are a number of complexes that will bind to these intervening introns. These happen to be made up of RNAs. And together they form an RNA machine called a spliceosome. And what they're going to do is cut out these introns, remove them, and splice together the remaining exons. In the end, what you're going to get is a much shorter, mature RNA composed of 1, 2, in this case, 3 exons. There will be a poly-A tail at the 3' end, a cap at the 5' end, and some regions before the start codon and after the stop codon that you can ignore. They are not translated. They have importance, but we're not going to go over their roles in this class. Note at the beginning of the first exon, there is a start codon, which is always AUG, which encodes for a methionine. And at the end of the third or the last exon, there will always be a stop codon. There happen to be 3 of those, UAA, UAG, UGA, any one of those 3, though I tend to just remember them with WAWAG-UGA, which just kind of rolls off the tongue and is something that is worth remembering if you get a chance. All right, so this is our actual mRNA, fully mature, fully processed, ready to ship out of the nucleus to the cytosol so it can be translated. Okay, except there's more. Now, we're not going to spend a lot of time on this, but not every gene is an instruction for making an mRNA, which is translated to make a protein. The other things to keep in mind is there are a bunch of genes for making things like tRNAs. While we have, all the tRNAs for the 61 codons, and that's probably on the order of 500-600 different genes because we have multiple copies of each of these. Then there are even rRNA genes. So to make ribosomes, you need 4 pretty sizable RNAs, and there are maybe 50 copies of each of those RNA genes. So that's like 200 more genes in your genome are for making ribosomes, plus the proteins that are part of ribosomes, which I forgot to mention. In addition, those splicing machines, the spliceosomes that took out the introns and put the exons together, those are actually RNAs, which means there have to be genes for them as well. And that's, over 1,000 genes that are involved in that process, 5 per spliceosome. And then in addition to that, and this is the last one I'll mention, there are interfering RNAs, RNAs that are used to target mRNAs and destroy them using specific nucleases in the cytosol. And so this might be anywhere from a few hundred to over 1,000. The data's on this, the data are very noisy. So it's unclear how many there are, and it's an area of a lot of contention. But let's ignore all that for now. The basic idea for most of the genes we want to focus on are they're transcribed to make an mRNA after processing, and we're going to hope that they get translated. So, to recap, for any particular cell type, most of your genes are inaccessible because they're condensed up. Of the ones that are accessible, a number of them don't have general transcription factors or don't have the appropriate activator transcription factors present at the right sequences, or there are repressor proteins preventing the transcription. If there's transcription available, that's the second level of regulation, then your pre-mRNA has to get processed, which we just talked about. Then, if that pre-mRNA is processed and the mRNA is made, it's got to get exported from the nucleus, which can be regulated. Then in the cytosol, it's going to have to get translated, and that's your ribosomes, which are made of our RNAs and proteins, plus the tRNAs that bring the amino acids. Then, we're still not done. Keep in mind that those proteins, those polypeptides that is, might get made at the ER, might go to the Golgi, might get some disulfide bonds added to it. Even if they're not in that pathway, the export pathway, they're going to have to fold up, and maybe multiple polypeptides will actually have to bind together in a quaternary structure in a giant protein complex or something of that nature. If you pass all these steps properly, uncondensed DNA, transcription factors present, RNA polymerase working it's doing its job, pre-mRNA being processed out of the nucleus, translated and modified, then you can finally get a protein. And per cell type, you've got a few thousand of these. Overall, we can, in a human, make more than 20,000. All right, that was both exhausting and shockingly overly simplified. Keep in mind the big picture. We're trying to regulate which genes in which cells at which times are on to properly determine a cell type and respond to the world as things are changing. Thanks, everybody. We'll see you next time.